Hello, my name is Chiffon Green. It is week 2 of microbiology. I am beginning on aseptic technique in culturing microbes. It is Wednesday, July 11th. I have my wick candle already lit here. Can't really see it, but I'm going to go ahead and get started on a few of the steps, and then I will bring you back into the video. Okay, so I have sterilized my pipette, so now I'm going to go ahead while holding it, and I'm going to sit here and sanitize my broth tube. And now I'm going to do the same thing for my E. coli too, my culture vial. And I'm going to go ahead and work on some more steps. Okay, so I am now getting ready to transfer my solution from my E. coli to back into my broth. And it says do our best not to touch the lids, or yes, the vial, just to not get any contamination. So I have done that, and now I'm going to do the same exact steps that I did for my E. coli for my S. epidermidis. Okay, so I'm now working on the S. epidermis. I'm going to go ahead and run the tip through the fire, and then I will do the same with the actual vial that the culture is in. And I'm holding the pipette because I've already sterilized it in the alcohol from doing the E. coli. Okay, so I have both of those sterile. I'm now going to take a bit of the broth and insert it into the tube, and then I will shake the tube and I will be back. I've successfully completed my cultures. So I have the yeast, which is the S. cerevisiae, and the E. coli, along with my S. epidermidis. I have to go ahead and incubate for 48 hours, and I will come back to you once I have that 48 hours passed and show you exactly what came about. Hello, so I am back with after the 48-hour incubation period. As you can see, I have all of mine here, all with growth at the bottom. Mine, to me, came out as the flocculent microbial growth that are at the bottom. They all have some at the bottom there, as you can see. So the growth did happen within that 48-hour period. I am now going to begin the second part of the experiment. So hang in there and I'll show you. So I'm beginning experiment 2. I have all my agar plates titled with their name on it, and close to them I have the samples out of the nutrient broth. I'm going to start with E. coli. So it tells me to go ahead and sterilize the top, run it through the flame. It tells me to open, and in one quadrant, after dipping this into the E. coli broth, it tells me to go ahead and make my quadrant 1. Okay, and it tells me to then insert my loop back in my alcohol, close up my E. coli broth, and then from there I'm going to do each quadrant. So I'm going to do quadrant 2, and then I'm going to continue with the other two to finish out the experiment. Okay, so I have all of my agar plates done now. I did the 4-quadrant sweep with the loop, and now I'm going to incubate for another 48 hours. I am back with exercise 2 of the aseptic technique in culturing microbes. My cultures have been in incubation for about 51 hours. As I have taken them out, you can see here we have the E. coli, the S. epidermidis, and the S. cerevisiae, also known as the yeast. So the next steps, it says to take pictures, so I'm going to go ahead and use my cellular phone to take the pictures. Kind of just going to use the sunlight to help with the photos, just to see a clear view. So I'm going to get those photos done, and then I will come back with the next. So the next step would be to, it says, after taking photos, include the incubation time and observations for the shape, and color. The E. coli is more of a yellow compared to the other two. I would say that's more of the translucent and white for the other two. My E. coli, I would say the shape is circular, some are irregular, if you can kind of see that. My margin, I would say entire to undulant. There's few of them looking like the two. Elevation is definitely raised. You can see, if you look through directly, you can see that the circular shapes are raised. And then the size is small, just a whole bunch of small circles. The yeast, yes, this is the yeast. I came up with the shape being irregular. The margin, I would say, is the filamentous. And the elevation, if you look straight through, it's not really elevated, it's more flat, but you can get some of it. There was one colony that is raised. And then I would say the size is large just because of this one right here. And then the last one I have is the S. epidermidis. For these, I would say it would be more of like a rhizoid shape, kind of all over the place. And then the margin, I would say, is lobate, kind of just squirt like up and down, up and down, up and down. Elevation is flat. If you look straight through, it's not risen at all. And then my size, I said large. So the next steps is just to insert my photos into my lab assistant, insert my observations for my shape and color, and answer my questions. So I hope you enjoyed the experiment of aseptic technique in culturing microbes, both my exercise 1 and exercise 2 through the incubation periods, and now the observation period. Thank you so much.